I'm Jim Hussey. I'm the CEO of Seneca Therapeutics. I'm a 40-year veteran of developing cancer drugs, okay? Which, by the way, is not always a really fun thing to do because you don't always work. So when I was at Bristol Myers in 1993, we launched this drug called Taxol, which everybody knows, and it cured 30% of the patients it was treated with. 30, remember that number. At the same time, lymphoma drugs that were on the market cured about 30% of patients, okay? So in the next 40 years or so, what we've watched is if you have lymphoma and leukemia, you won the cancer lottery because you're going to get cured from your tumor and you're going to live a long and robust life. If you have a solid tumor, which is 80% of all people with cancer, your response rate is 30%. Isn't that great news? How many of you have seen the Keytruda ads on TV? Aren't those amazing ads? Give Merck a lot of credit. Yeah, the response rate for Keytruda is 30%. Okay? So what happens is, what is it about solid tumors that's different than the blood-borne tumors like lymphoma and leukemia? Well, one thing is different, and I was ready to retire in 2020, and I was approached by these guys, and I just launched Monjuvi, which is for diffuse large B-cell lymphoma, and if you happen to take Monjuvi, you have a 95% cure rate, same as you've had strep throat. But these guys came in and said, we think we understand why solid tumors don't respond. And it's a little marker called TEM8. And I said, oh, that's interesting. Let's go prove it. Because remember, at the end of the day, there's lots of things in cancer they had tried, and very few of them work. So let me talk a little bit about this, where we are. So Seneca Therapeutics is built on 2 years of work, or 20 years, 2 decades of work. One of the truths about when people tell you that there's quick answers to things, don't believe them. Do not suspend disbelief. Most cancer breakthroughs are essentially hidden for 20, 30 years until sometimes 2 or 3 things come together. So what happened was Seneca Valley virus was an accidental discovery. It was a contaminant in a fetal bovine serum sample at Novartis. They happened to notice it killed some cells, but not other cells. They went out and they tried it. They did it in some clinical studies. They didn't know how it worked, right? And so essentially it worked in some patients, not others. They dropped the drug. This was a company called Neotropics. It was in the early 2000s. There was a guy at the National Cancer Institute named Brad Sincroy who discovered TEM8. It was the same marker, by the way, that sits on embryos that happens to defend the embryo from the mother's immune system. Surprise, surprise, he finds it in cancer cells. Clue, there's a clue. What are cancer cells and tumor cells share? They're both fast growing. They both have to have immunosuppression in order to grow because your immune system will attack them, right? And they have to go find a better food source. Now, for the baby, it goes to the placenta. In a tumor, it goes and metastasizes. And where does it metastasize? The liver, the lung, where there's lots of blood, right? So that's how those things work. First thing that was interesting. So TEM8 comes along. There was 3 clinical studies back in the day. And then all of a sudden, this guy named Charlie Rudin in 2017 at Memorial Sloan Kettering finds out how TEM8 works. And he finds out that Seneca Valley virus is the only thing that binds to it. Seneca Valley virus, the contaminant, it turns out it's a pork virus. It doesn't do anything except bind to and kill TEM8 cells. It wasn't engineered. It wasn't discovered, right? It was there. It's a pork virus. It doesn't even cause illness in pork, but it's there and it kills these cells. So this is the story I'm going to tell you. So all of a sudden, people started looking at TEM8. And here's a great question. If overexpression of TEM8 is the problem, shouldn't you see a reduction in survival in people that have high TEM8? Wouldn't that make sense? If you have lots of TEM8 on your tumor, you should live shorter lifespan than if you have very little, right? So back in 2011, we started looking at this. Well, surprise, surprise, we said, how about small cell lung cancer? Well, if you have a high expression of TEM8, you live 20 months. If you have low expression, you live 34 months. And that's with standard of care. The only thing we looked at was one marker. We didn't look at a whole basket of markers. We didn't look at a liquid biopsy. One marker, okay? We did the same thing with bladder cancer. It was 27 months, and those are all statistically significant, clinically relevant. 27 months versus 45 months. And then we turned around and said, how about in things like cervical cancer? Well, essentially it was 67 months survival if you had high expression, and it was infinity if you had low expression. Who wants low expression on their tumor? Yeah, everybody raise their hand. What we're trying to do by killing the TEM8 positive cells is to make high expressing tumors into low expressing tumors or no expressing tumors. Okay? Then we went in and we said, how about triple negative breast cancer? 200,000 people a year. It's one of the most deadly forms of breast cancer, okay? Well, look at this. Same thing happens. If you look at triple negative breast cancer, 50% of the patients with high expression live about 3 years. Golly, the people with low expression, they're going to die at 90 in a nursing home, right? Same disease, but one has high expression of TEM8, one has low expression. Same is true in gastric cancer. We looked at 12 different tumors. Every single time we found high expression of TEM8, the patients lived a statistically and clinically relevant shorter lifespan. So what's the problem with solid tumors? It's TEM8. But nobody's thought about this, right? Nobody's really thought about this way. We all have developed drug for the 30%, right? Hey, we got a brand new drug and we have a 30% response rate. And we have a whole new class of drugs that are 30% response rate. That's the world I grew up in. That's what I've been dealing with for 40 years. People would come here, tell you they have a new drug, and you ask, well, what's the response rate? Oh, we're at 32. Oh, wonderful. So that makes those 2% of the people happy. That's not going to change solid tumor cancers. 800,000 Americans die every year of solid tumors. Now, the question is how broadly, this is a picture of this little sucker, okay? So the thing in the middle is Seneca Valley virus. The stuff on the outside is TEM8. Well, it turns out TEM8 has 60 binding sites, which is essentially undruggable, right? You could never get a drug that bounds 60 places, right? We're lucky we have 2. A monoclonal antibody, state-of-the-art, binds to 2, all right? But for some reason, I always tell people, God designed Seneca Valley virus. God designed TEM8. We didn't. We just discovered it, right? So we look at this thing and you look at it, it just completely surrounds the virus. It was like TEM8, they call it a lock and key. It was made for each other. And what happens is when Seneca Valley virus goes inside of a TEM8 positive cells, it makes 200 to 400,000 copies. It bursts the cell from the inside. And then guess what happens? It releases more Seneca Valley virus to kill the tumor next to it, and the one next to it, and the one next to it. So the crazy part is, if you were to give, an initial dose of Seneca Valley virus, it actually is 5 to 7 times higher in day 5 because the tumor is acting like a factory for the virus that's killing it. Kind of fun, right? You couldn't design that stuff if you made it up. That's been the problem, okay? You can give all the standard of care, but if you've got a high-expressing TEM8 tumor, it doesn't matter. Drugs don't work, chemo doesn't work, radiation doesn't work, and it metastasizes on top of that because it thinks it's an embryo, right? That's what TEM8's doing. It thinks that tumor is an embryo, and it's making the tumor act like an embryo, all right? So all 7 known cancer cell types express TEM8. So we thought, well, maybe we get lucky and there's one cancer cell type. No, they all did. All of this is brand new, all right? So then you go back and look at, well, how common is it? 65% of all solid tumors express TEM8. Wow, does that sound like 70%-ish number? Remember that number of the solid tumor drugs that don't, what percent of patients don't work? 70%. Wow, 65 sounds kind of close to that. But what's really hard is it's not the same across tumors. You get tumors like glioblastoma, it's 100% of those tumors. Sarcoma is 86%. Other tumors, it's lower, okay? So what you got to have is a companion diagnostic. That's one of the things we had to develop for FDA when we got the IND approved, okay? So a patient has to know, we have to know that that patient has a TEM8 positive tumor. Because if you give Seneca Valley virus to a patient who doesn't have TEM8, you're wasting your time. It won't do anything. But if it does, it makes a huge difference. Now, so I'm going to go this slide first. So these were crazy studies. So we went out and we did this mouse model. Essentially, we've done it in humans as well. I'll talk about that. But essentially, you get this normal mouse. And essentially, if you treat it with Seneca Valley virus plus 2 other drugs, 100% eradication. This PAN02 is a pancreatic model. Most other drugs don't work, right? So what's crazy about it is this one, if you it wipes out the tumor and it stays wiped out after the drug is gone. Because it's using the immune system. If you take away the TEM8 cells, the immune system says, oh, that's a tumor, I'll go kill it, right? But while the TEM8's sitting on there, it's going to defend that tumor from all the drugs and radiation and chemo and everything else you could think of. At the same time, we went back then on day 26 and we re-challenged with a tumor on the other side of the body, right? And guess what happened? The animal killed it without doing any more drugs, any more therapy. It destroyed the tumor because the immune system suddenly recognized it because TEM8 wasn't there. It suddenly recognized that tumor for what it was, and it killed it. Those mice, all the other mice were dead by day 90. At day 165, we finally sacrificed the mice. There was literally nothing there. All the animals were healthy and they were doing great. So with that, I've got the walk-up here from Miss Brianna, so I'm done. The rest of it is very quick. I can go through it. There was lots of good data. There's a study starting at the University of Miami. They're paying for it, which is pretty amazing. And they're essentially looking at phase 1/2 trial. It's all set to go. Well, first patient will be enrolled in November. And essentially, we're raising $10 million to do our part of that trial. Thank you guys so much for listening to this story. I'm very excited. After 40 years of bad news, maybe we'll have some good news.